Okay, this is chapter 22. We're going to be taking a look at DNA and biotechnology. So, first of all, we've got to talk about DNA. Right, so DNA basically is deoxyribonucleic acid. And so this is our genetic material. And so your DNA is organized into genes, which are basically short segments of DNA that have instructions for specific traits, and they're organized into chromosomes. And all eukaryotic cells, living things have DNA. And in eukaryotic cells, that DNA is located in the nucleus itself. And there's a little bit of DNA also found in the mitochondria, but most of our DNA is in our nucleus. Now, DNA has three main functions. First of all, its job is to replicate so that it can basically transmit to the next generation. It also stores information, so it's an informational molecule. And it can also undergo change, mutation, and that gives you variation. So the structure of DNA is actually fairly simple. It's a double helix. It's like a twisted rope ladder. It has two strands that basically spiral about each other. And it's composed of a series of nucleotides, which are the subunits for the DNA. And those nucleotides basically have three main pieces. It has what's called a phosphate group. It has a sugar group. And in DNA, it's deoxyribose. And you have one of four possible nitrogenous bases: adenine, cytosine, guanine, or thymine. Now, the adenine and the guanine are basically called purines. They have two rings on them, and the cytosine and thymine are pyrimidines, which have one ring. This is kind of what it looks like. So you can see you basically will form your phosphate sugar backbone, this repeating unit as these phosphates and sugars are connected to each other. And then sticking out to the side, like the rungs of the ladder, are going to be the nitrogenous bases. And those are going to be paired up with each other with the other strand. And that's basically kind of what gives you the rungs of the ladder. And then if you just give this a twist, then you basically get your little twisted rope ladder. Now, the backbone, which is your sugar phosphate backbone, are kind of like the rails of the ladder themselves. And then the base pairs are complementary. They're paired to each other in a complementary fashion with hydrogen bonds. For instance, A's and T's will pair together with two hydrogen bonds. And C's and G's will pair together with three hydrogen bonds. Now, when you take a look at this, you can see you can break it all up. So you can see your phosphate, which is going to be on what's called your 5' end. That's just associated the designation of that carbon that it's attached to. On your 1' carbon is going to be your nitrogenous base. And then on your 3' carbon, which is another important carbon, is going to be an OH group. And so ultimately, this will form your nucleotide. And then, of course, they just basically pair up with each other, forming hydrogen bonds to kind of zipper up to form the other half of DNA. So the two strands of DNA are important. That basically means they're anti-parallel, which means they kind of run in opposite directions. So on one side, you have the 5' end, and that leads to the 3' end. On the opposite strand, it goes the opposite direction from 5' to 3'. And so that's kind of like the complement of that. And so this is because ultimately of the way the carbons are positioned and the kind of the polarity of where each of these pieces are, right? For instance, you have a phosphate at the 5' end, but you have an OH group at the 3' end. And so that kind of creates that sort of northbound, southbound type of antipolarity that you see that. Now, DNA replication, which is essentially the process of copying your DNA, is something that happens in S phase of the cell cycle, which we talked about a few chapters ago. And so each of the DNA strands will act as a template that essentially allows you to make a new strand that's complementary to that template. And so this is a process that is what's referred to as semi-conservative. Essentially, you have any given DNA molecule, you have one new piece and one old piece or one original strand. So you have two different strands, a new strand and an old strand. That's kind of what it looks like. So basically, when you go to replicate your DNA, you essentially unwind your DNA, and then the old strand acts as the template, and then you make the new strand based off of that. So your new DNA molecule is actually going to be a hybrid of old and new strands. And this is what's called the semi-conservative model. So that's basically the process of DNA replication. Now, there's a couple of different enzymes that are in play when it comes to DNA replication. First thing you have to do is you have to unwind the DNA to get access to that template so you can read it. And the enzyme that does this is called helicase. Oftentimes I refer to this as the unwindase. And so this guy will unzip the DNA or basically open it up. And that exposes the bases of the template so that the polymerase, which is the next enzyme in line, can read it and basically make the new strand. Now, the DNA polymerase is essentially the one that makes the new strand. So this basically adds all the new DNA nucleotides based on those complementary rules, and it essentially makes the bulk of the DNA. So when you make that new strand, it's DNA polymerase is doing it. Now, because of the polarity of the DNA, synthesis typically occurs in opposite directions. Why? Because that's due to the polarity and the anti-parallel nature of DNA. Remember, the important thing to understand is that polymerase itself can only add nucleotides to the 3' OH. So it has to have that 3' OH. Where's the 3' OH? It's only on one end, right? You got your 5' phosphate on one end. You have your 3' OH on the other end. So the new polymerase can only add new material to that 3' end. That basically means it has a 5' to 3' directionality. Because of that, that basically means that each strand is going to be going the opposite direction. And that breaks these two strands into two different types of strands, one called the leading strand, the other one called the lagging strand. And the lagging strand basically will create these small DNA chunks or fragments called Okazaki fragments. And so we'll take a look at this. So on one side, we have the leading strand, where essentially a DNA polymerase gets a hold of the template, and because the 5' to 3' direction is moving into the replication fork, it can just basically proceed without stopping, because as soon as the helicase opens that up, it basically allows the DNA polymerase to just basically move right on through and copy the template that's newly available to it. However, on the lagging strand, which is this guy over here, your directionality is the opposite direction. So your 5' to 3' direction is going away from the replication fork. So now what that does, that basically means that the DNA polymerase has to basically move away from the replication fork. So not only that, but you need a little bit of a starter. So you have a bit of a primer right here. So basically a short stretch of RNA that provides a 3' OH for the polymerase to build off of. And when the polymerase lands on that, it can basically fill in everything ahead of it until it runs into an obstruction, like, for instance, the primer that's just ahead of it. The problem is all of the new template that needs to be copied is now behind it. So once the polymerase gets to this point, it has to fall off and has to come back and rejoin behind it. And that's the reason why they call this the lagging strand, because it kind of has to keep falling off and then jumping back and then continuing through. Now, ultimately, once all of this is completed, you can see there's other enzymes that'll come in here and that'll replace the primer with DNA. And then a DNA ligase will come in here and seal that nick. So this basically seals the nicks in the backbone and essentially creates the full, complete DNA molecule. So a mutation is essentially an error in replication. And so if you kind of do a mismatch or a mispairing, then that would be a mutation. We have enzymes that can fix that. So entire groups of repair enzymes can go in there and fix the mutation or fix the damaged DNA. However, they can't always fix it. And so most of the time, mutations are a disruption in the gene and it's a negative. But every now and then, you might be able to get a beneficial one that could maybe add new variety to you. Like, for instance, the smash face look of a Pekingese, for instance, is the little smash face dogs. That's a mutation in normal muzzle development. And normally that would be a negative, right? Because the dog has lots of issues with drainage and things of that nature. So probably in the wild, that would be a negative. But for us, because we think it's really cute, it turns out to be a benefit to the dog. So that's kind of an example where every now and then a mutation come along that actually provides a bit of a benefit. So that's DNA, but there's another nucleic acid we want to talk about. That's RNA, ribonucleic acid. And so this basically is also made up of nucleotides. However, it has a different sugar. It has a ribose, and it has also 4 nitrogenous bases, adenine, cytosine, guanine, but in this case, instead of thymine, it has a uracil. So it also has the same complementation. So for instance, you still have your C with G, and A's in this case will pair with U's, but by the same basic rules. However, RNA is usually single-stranded, so you don't really use the base pairing as often as you do with DNA. So here's your RNA molecule. You can see it up close. You still have your phosphate sugar backbone. And sticking out to the side, you can see your nitrogenous bases, where you have uracil instead of thymine. So this is a table of some of the differences between DNA and RNA. For instance, the sugar is uniquely different. So deoxyribose relative to ribose for the RNA. The bases are different. You have uracil instead of thymine. They're double-stranded in DNA, single-stranded in RNA, and so forth. So this is a good table just to kind of give you an idea. The difference between the 2 different nucleic acids. Now, there are different types of RNA. There's actually multiple types of RNA, but this is kind of the standard 3 main types. The first one is messenger RNA. Then we have ribosomal RNA, and then we have transfer RNA and other types of RNA, small RNAs. The messenger RNA is essentially the one that is produced in the nucleus by a process called transcription. And this is basically a copy of the DNA. And what its job is to do is it'll take the information from the DNA to the ribosome so that you can make a protein. So this is basically carries the messenger RNA, carries the code that is embedded in the DNA for your amino acid selection. And that's going to be in protein synthesis. Now, ribosomal RNA is also the same way. So this is going to be, you're going to find this aggregated in those central structures in the nucleus called the nucleolus. This is actually a structural component of ribosomes. So you'll see this basically forming with both the large and the small subunits of the ribosome. And so it'll leave the ribosome, and then it'll form the structural component. So the ribosome is actually a component of both protein and ribosomal RNA. And then transfer RNA, this is also produced in the nucleus by transcription, same process. All 3 of them are transcribed. But what this does, basically, it connects the idea of amino acids with that of the genetic code. So for instance, it's here at the tRNA that you get the designation for a particular type of DNA code, giving you the assignment for a specific amino acid. For instance, the DNA code that is AAA will transcribe into an mRNA that is UUU, and then that will be taken by the tRNA, and it'll translate that into an amino acid selection, which is, in this case, phenylalanine. And so each tRNA has a specific amino acid. And so there's about 20 or so different tRNAs. Now, some of the small RNAs are basically little chunks of RNAs, and so we have different types of things. So small nuclear RNAs, for instance, snRNAs, these typically are involved in a process called splicing. You only see that in eukaryotes. Small nucleolar RNAs, these will modify ribosomal RNAs within the nucleus. There's also small microRNAs, little pieces that are out throughout the cell, oftentimes have a regulatory role. So these guys will oftentimes have a regulatory type of a role. In the cell. They're not necessary for translation, but they can regulate different things. Small interfering RNAs can also bind to RNAs, but they can designate these RNAs for degradation. So if you don't need that RNA anymore, then you can use that small interfering RNA, the siRNA, to basically go in and break that mRNA down and kind of get rid of it. So you don't just have it hanging around all the time. So let's go ahead and take a look at gene expression. So gene expression is an idea that can be thought of as, and you can think of this way. So the manifestation of a gene, how a gene manifests itself in the phenotype. That's gene expression. So ultimately, gene expression and genes generally will express themselves through the protein that they make. And so that means you're going to need transcription in order to make the RNA, and you're going to need translation in order to make the protein from that RNA. So once you actually have your proteins are going to be composed of amino acids. There are about 20 different amino acids. And so they differ in number and order. And so the sequence is basically given the protein its characteristic. It's also determined by the sequence of what's in the DNA. So this will determine the structure and the function of various things in your body. And so you have different types of proteins. So you basically have structural proteins, you have regulatory proteins, you have enzymes, which are catalytic proteins, neurotransmitters can be proteins, antibodies are proteins, hormones can be proteins, for instance. So you have a lot of different functions of these proteins. There's a lot of diversity. And that's because you got a lot of different ways that you can arrange these 20 different amino acids, different sequences by which you can select them. And all that information is housed in the DNA itself. Now then, let's go ahead and take a look at gene expression. In order to understand gene expression, we have to understand the two fundamental mechanisms that underlie gene expression, which is transcription, which is the first step of gene expression, and translation, which is the second step. So in transcription, basically what's going to happen is you're going to take your DNA, which is sort of your ultimate source, your original hard copy, if you will, your blueprint for all the information that you need. In this case, I would just want to make a protein. And so the information for how to make that protein is embedded in DNA. But since I don't want the entire DNA genome, that is every single gene that you have, I just want my gene. Then what I'm going to do is I'm going to run off a copy of that sequence in the form of mRNA. That's your transcript. And then from there, I'm going to take it to the ribosome, and I'm going to have that mRNA translated into amino acid sequence, and that's the process of translation. So if you take a look at it then, here you have your DNA strand. Only one strand at a time is typically transcribed. And so generally it's transcribed in 3-letter units called codons. So these are the 3-letter words of transcription. So for instance, this would be one amino acid here. And this would be the second amino acid. This would be the third amino acid. So you have 3 codons, 3 amino acids. And so basically running that copy off from the DNA gives you your mRNA. And then this goes to the ribosome, where the tRNA will connect that amino acid that I want and start to connect it in a series, which becomes our polypeptide. And then we can kind of go back to our previous chapter earlier in the semester where we talk about protein folding. So once we actually have that linear sequence of amino acids, then we can connect that to the protein folding mechanism. And then after that, we'll get our functional protein. Now, the big question that comes from all of this is the idea of the code, right? So for instance, how does a genetic code actually work? So if the information in the DNA, which is essentially just a series of A, C, Gs, and Ts, is everything you need in order to confer the information you need for making a protein, then the question is, how do you encode for that protein? So there's this issue of genetic code. So the bases in the DNA will encode the amino acid. Of course, we already talked a little bit about our codon, which is our 3-base sequence in mRNA. Basically, each one is an amino acid. That gives us about 64 possible codons, of which 61 of them actually correspond to a specific amino acid. The other 3 are what's called stop codons. And so this is important because you have to know when to stop your translation process. And of course, then one of them is AUG, which is called a start codon. So basically, this is your start point. So essentially what you have is you have a start, you have a bunch of codons in between that'll become amino acids, and you have a stop. Kind of like a sentence, right? With a sentence, you have a capital letter that forms the beginning. Then you have a continuous reading frame in the middle until you come to the stop, which is your terminal punctuation. Okay. So this piece right here, this frame that you're reading is oftentimes what's referred to as an open reading frame, or an ORF. So this is what our genetic code looks like. So for instance, if I were to take a piece of sequence, and I'm going to do that now, let's say I have, let's say AUG, GGG, UUU, UAG. Let's say I have that. If I want to translate this, then basically the way I do that is I take my first, break them up into codons first. So I have 4 different amino acids possibly that I'm going to be using here. So the first one, that is A, is going to be in this column here. And so I can just go and find my A. So here's my A cluster there. And then the second one, which is going to be U, is going to be found along, second ones are going to be found here. So you can see the second base is U, so it's going to be in this column. So where these 2 columns intersect, that's basically where it's going to be. And my third one is a G. So in this case, I've got my start codon. AUG is methionine. So that first one would be methionine. Now, if I want to see GGG, I go down to the G's. I have to go all the way over here to the other G's, and I can see there's GGG right there. It's glycine. So this will become a glycine. And then if I want to take a look at UUU, so here's my U's right here. These are also my U's. So there it is right there on the top. So that's a phenylalanine. And then a UAG. So here's my U. My A is over here. Notice UAG is a stop codon, which pretty much means I'm done. There's nothing there. So what I have is methionine, glycine, and phenylalanine in a series. That's basically how you use that table. So a lot of enzymes have what's called redundancy. They have more than one codon. So for instance, if you take a look at serine, right, you can see that serine has at least 2 different codons right here, AGU and AGC. Leucine has a whole bunch of codons, right? So you can see all these different codons will encode for leucine. That's called redundancy. Now then, when you take a look at the mechanism of transcription, a piece of the DNA or a segment of the DNA, one strand, will serve as a template for the production of the RNA molecule. And then ultimately, all of your classes of RNA, regardless of what it is, whether it's mRNA, rRNA, tRNA, or whatever, is going to be formed by transcription in this process. The first thing we have to start with is our RNA polymerase. This is the enzyme whose job it is to add the RNA nucleotides to the complementary, using these complementary base pair rules. For instance, in this case, it swaps out a uracil for the thymine. So this is kind of what it looks like. So here you can see the RNA polymerase is unwinding the DNA up here. And then as it goes through, it's going to basically pair off. So it's going to add an A right there to the T, a C there, and a G there. And it's going to keep on going. And it's going to keep on growing this piece out. So it's going to basically kind of keep going out like that. And so essentially what's happening is you're getting this kind of directional movement, and you're essentially making a complementary copy, a single-stranded copy of that one piece of DNA strand, that one DNA strand. So once you actually get your mRNA out after transcription is done, then you're not quite ready as a eukaryote to go out and start making protein out of the information you just copied, because your mRNA has to be processed. At this particular point, what you have is what's called premature RNA, right? Or pre-mRNA. You still have this 5' end and this 3' end. But part of the problem is that in the middle of it, you also have a bunch of different things. So you'll have different areas that are called exons. Exons basically are portions of this particular transcript that are expressed. That is basically, they become protein. These will actually tell you to pick an amino acid. And so you'll have multiple exons in this particular region, different genes are different. But then in between them, you'll have introns. And introns basically are other areas that disrupt or interrupt the gene. They don't code for protein, so they're not coded. They don't become part of the protein. So you got to get that out. Because remember, what you want is you want to be able to start somewhere, have an open reading frame, just basically read through it, and then have a stopping point, right? And that's what you want. Right now, you don't have that. Right now, you have all these interruptions, all these intervening pieces that are in between it. So you got to remove those to create a nice, continuous, open reading frame. And so this primary or premature mRNA has to be processed. And one of those things you have to do is basically snip out those introns. And so those introns basically are snipped out by a process called RNA splicing. For instance, one thing you have to do also, let's just kind of put it all together, is because you have a single strand, right? This is single-stranded RNA. Remember, the destiny of all single-stranded RNA is to be degraded. So essentially, if you have single-stranded nucleic acid out there, it'll get degraded. If you don't want it to get degraded, you have to protect it. So for instance, you have exonucleases which will come in here. They'll see this 3' end, and they'll basically want to chomp on that 3' end and devour it. You also have other exonucleases which work on the 5' end, so they'll want to chomp on that 5' end. So what you have to do is you have to process your mRNA to block that degradation. And that's exactly what we start doing. So basically, what we do is we put a cap, there's sort of a 5' cap, that basically protects this mRNA from degradation at the 5' end. And you also put a poly A tail on the other side, a series of adenines that has the effect of blocking those other exonucleases. So you protect them as well. The next piece that you want to do is once you've capped it and tailed it, then what you want to do is splice out all the introns. So the process of splicing is essentially the removal of introns. And so you have to identify them first of all and then cut them out. And so there's a complex of proteins, and actually they're proteins and RNA, snRNA actually, this is where the snRNA is involved, called the spliceosome. And the spliceosome's job is to recognize these introns and then ultimately cut them out. And so the RNA piece acts like an enzyme here, so it's catalytic. And when you have a catalytic RNA, we call it a ribozyme. And so through this particular process of splicing, what's going to happen is you cut out the introns and you sew the exons together. So for instance, you can see that process here. So here you can see the spliceosome is identifying this intron here and here and here and here. It's going to cut those out, and then it's going to sew the exons together. So here you can see you've cut out the introns, you sewed the exons together. Now what you have is an ORF, an open reading frame, with your start site here and your stop site here. So just like a sentence, right? And so that's important. Now, you can also, from this, create what's called alternative splicing. So for instance, some cells can basically swap out different exons based on how they cut. And so if you take a look at this, just to give you an example of what this kind of looks like, let's imagine we have, here's exon 1 with an intron, a little exon 2, and so we have exon 3, and exon 4. Well, one of the things that happens is you basically tend to nip out each of the introns and you sew the exons together. So what you'd end up getting is exon 1, exon 2, a longer exon 3, a little bit of a shorter exon 4. But what you can do is you can actually nip out different patterns. For instance, what if I wanted to cut all this out like that? Then what I would end up with is basically exon 1 and exon 4. So a different open reading frame. And so you can use this to sort of get different types of protein products out of that. It's kind of like an abridged book, basically. right? It's not the full thing, it's just an abridgment. Okay. So now once you've processed your mRNA, you've capped it to save the 5' end, you've tailed it to save the 3' end, you've cut out the introns so that you have an open reading frame. Now you're ready to export throughout the nucleus and head to a ribosome for translation. This is where your tRNA is going to be bringing the amino acids to the ribosomes to be incorporated into the protein. So the translation complex is essentially the ribosome, which is a component of two major subunits, a large and a small subunit, with its ribosomal RNA, and it's going to bind to mRNA. And when it does this, it creates three different active sites called the A, P, and E sites, where essentially the individual tRNAs will bind to the mRNA. So this is kind of what it looks like from the outside. So you have your large subunit basically on the top, your small subunit on the bottom. You'll have your mRNA kind of threaded throughout there, and your polypeptide kind of growing as it basically adds amino acids in response to what's on the mRNA. So this is what the EP and A sites look like. So the A site typically is what I refer to as the arrival site. This is where the new tRNA with the new amino acid that corresponds with this sequence, GAC, comes in and basically lands in that A site. That's the arrival site. The P site, which is this guy right here, this is what I called the peptide site. This is the site that carries the peptide chain that you're working on. And then the E site, this is the exit site. So once you transfer your peptide to the growing chain, you're empty now. And as a tRNA, this is where you exit. Okay. So on the tRNA itself, it's got two different main areas for binding. The first one is what's called the attachment site for the amino acid that it has. And then the other side is an anticodon, which reads the mRNA. And so just like the mRNA codon, the anticodon is also a 3 base pair sequence, but in this case, it's complementary to the mRNA codon. So for instance, if you have your DNA that says it's AAA, your mRNA will make the complement to that, so it'll be UUU, but guess what the anticodon is? The anticodon would be AAA, because it has to be complementary to read that mRNA. And so the tRNAs that have their amino acid will go to the ribosome and they'll attach at the A site. That's the arrival site. And then the amino acid they're carrying will essentially be the one that adds to the polypeptide chain. So in this particular case, you see CUG. This is your anticodon region down here. And then at the other end, you have your amino acid. In this case, it's aspartate. So the order of the tRNA delivery is associated with the mRNA codons. So what codon, the codon basically determines which tRNA comes and binds to the A site. And so once it's bound to the A site, then it'll be able to transfer that amino acid to the growing chain. So the stages of translation then are, there's three main stages. There's initiation, so events to kind of get you started. There's elongation, that is basically reading through the transcript. And there's termination, that's basically a stopping point. And that's those three. So for translation initiation, this is basically where you set things up. So the mRNA will bind to the small ribosomal subunit. This will basically form that translation complex, right? The ribosome itself. So that's kind of what it looks like. So you can see, first of all, to the small subunit, you're going to have your mRNA binding to it. You're going to have that first, what's called initiator tRNA in line, read its codon and kind of get itself in place. So now you have these three attached. And then what happens is the large subunit will come in and cap it. And when it does, it'll create the three active sites, E, P, and A, with the current amino acid, the initiator tRNA, in the P site. So now you're ready to accept the new tRNA as you read the A site. Now during elongation, this is where your polypeptide will start to elongate, where you add amino acids one at a time to the growing chain. So the incoming tRNA will arrive at the A site. And so at this particular point, what's going to happen is your P site is going to be filled with the tRNA that has the peptide complex, the growing peptide chain. And then in the E site, you have your spent tRNA, which has given up its peptide. That basically will exit. And that'll basically allow you to be open up the next that'll basically allow you to be able to open up the next A site. And this is kind of what it looks like here. And I just have to pause real quick. I'll be right back. Okay. So ultimately what's going to happen, the first thing that's going to happen during elongation is you're going to get what's called codon recognition. And so what's going to happen here is the anticodon is going to come in and it's going to read its little 3-letter codon code. It's going to lock into place there. So you can see in the P site, you've got the tRNA with the growing polypeptide chain. Then what's going to happen is you're going to move the chain over and you're going to attach it to the new amino acid. And so at that particular point now, notice the tRNA that is carrying the polypeptide chain is now in the wrong site. It's in the A site. So what has to happen is now what's called translocation. So translocation basically is going to pull the mRNA this direction. That's going to shift these tRNAs over one. So the empty tRNA will enter the E site where it gets ejected to go get recharged. It has to add its amino acid back there. And then this other one is going to be shifted back into the P site. That's going to make the new codon available so that your new tRNA can come in and then add its amino acid. So you go through elongation until ultimately you hit termination. And so termination essentially happens when one of your 3 stop codons hits the A site. And then what happens here is instead of a tRNA coming in, you're going to have a release factor coming in, which is not associated with an amino acid, so it just basically stops. And then everything just gets released. So the ribosome will dissociate, it will release the mRNA, it will release the polypeptide, and then from there on, your polypeptide has to go through those 4 stages of protein folding, and then your ribosome can just basically reassemble around another mRNA, and you do it all over again. So a polyribosome basically is a situation where you have multiple ribosomes on the same mRNA. Because after all, if you've got the mRNA available, and it's a fairly long one, you can fit more than one ribosome on it, why not, right? And so you'll have lots of different polyribosomes on one RNA. So basically multiple ribosomes happening. So this is kind of what it looks like. So in the DNA or in the nucleus, you have transcription occurring, right? You open up your DNA template, your polymerase will make a copy of that. And then once you make a copy of that, it's premature RNA. So it needs to be processed, and you're going to process that. You're going to cap it, tail it, and splice it. And you're going to move that RNA out of the nuclear pore into the cytoplasm where it's going to be bundled up with a ribosome. And then you're going to go through your initiation, elongation, and termination associated with that translation process. So this is just basically a nice little table to help you organize the different participants of gene expression to kind of put together the components we've just been talking about to kind of put them together. Okay. So how do you regulate this? So this is gene expression, that's transcription and translation. Then how do you regulate this, right? How do you control it? Ultimately, all cells have all genes, but not all genes are active. And so different cells will have different genes that are expressed. And so there's a lot of different ways to control them. One way to control them is what's called pre-transcriptional control. So you can uncoil the DNA, packing it tighter so that enzymes can access transcription. That's one way to do it. You can also modify the DNA to protect it so that it's not transcribable. So you can change the access of the DNA. So if your DNA is not accessible, then you're not going to be able to do anything with it. Another way is called transcriptional control. So this is basically a situation where you can regulate the transcription genes that are responsible for doing the transcription. You can do this with what's called transcription factors, which are essentially helpers in the transcription process. And they can have both inhibitory and excitatory or roles. So they can either enhance transcription or they can depress transcription. And then you have what's called post-transcriptional control. This is basically, you've already done transcription. This typically will happen in the nucleus after the DNA is transcribed. And this is before the mRNA is formed, right? So for instance, post-transcriptional, an example of this would be alternative splicing. That would be a type of post-transcriptional control. And then, of course, you have translational control. So this occurs in the cytoplasm after it leaves the nucleus. For instance, some of the ways that you can regulate this is those micro mRNAs. So you have the mRNA there, but a microRNA can bind to it and inhibit its translation. That's a type of translational control. The small interfering RNAs as well, they can prevent translation by marking the mRNAs for destruction. So that's another way to regulate translational control. Just because you have the mRNA made and ready doesn't necessarily mean you're going to translate it. And so there's translational control that can do that. We also have post-translational control. So post-translational control is once you've made the protein. So basically, you've made the protein. Now we modify it. So for instance, an example would be a glycoprotein. In which case, adding a carbohydrate, it would be considered a post-translational control mechanism. So this is kind of what it looks like. So here you can see you can have pre-transcriptional control, regulation of accessibility of DNA. You can have transcriptional control, how you regulate the transcription of a particular product, post-transcriptional control, how you're modifying that mRNA before it leaves the nucleus, translational control, and then post-translational control, where you can add things to that particular protein. So that's basically a good that's kind of a good strategy for control. Now we're going to take a look at transcription factors here in just a second. Okay. Back to business. Now transcription factors basically are other proteins. They're usually DNA binding proteins, and they'll kind of help set up this process. So they tend to bind to DNA. For instance, polymerase, RNA polymerase will attach to DNA and it begins transcription. And so this basically will allow you to sort of control transcriptionally how transcription works. Of course, as cells mature, they differentiate, they become specialized for specific tasks. And so oftentimes the specialization, which we call differentiation, is associated with which genes you're turning on and which genes you're keeping off. So let's go ahead and take a look at some biotechnology. So with this one, basically what we have is a situation where we use systems to create a product or achieve some sort of other end. For instance, we're trying to, like a therapeutic or something like that. This oftentimes will involve genetic engineering, which is essentially the modification of a genome, either to improve their characteristics or to get us the biotechnological product that we're looking for. Oftentimes we'll be using mostly bacteria, plants, sometimes animals, but mostly bacteria and plants. And this basically will allow us different types of techniques. So we learn different techniques so that we can do things like cloning genes or directly edit the genome of an organism. So these are all things that we can do. One important technique that we do is called DNA sequencing. So we can essentially determine the order of nucleotides in DNA. The important thing about this is it allows us to be able to determine what the alleles are, especially as they're associated with a disease. So like if you have a disease gene, like a genetic disorder, we can sequence that gene to figure out what's going on. Maybe that'll give us some information to help us to maybe kind of treat that particular genetic disorder. This will also help us in forensic biology. For instance, we can sequence your DNA to determine whether or not you were at a crime scene. Or we can actually sequence DNA that we find, say, for instance, in a fossil or something of that nature, which we have been finding, not necessarily DNA, but we've been finding soft tissue in fossils, which is an amazing thing in of its own right. But if we ever find DNA, we could do sequencing. We've done that for instance, with the woolly mammoth, right? So we found, pieces of mammoth that's had DNA in it. We've been able to sequence that DNA. And so ultimately, when we sequence the DNA, we want to make many copies of it so we can understand it. We use an amplification process called polymerase chain reaction, which is very similar to replication, and it has three main steps. So the first step basically is to denature the piece of DNA. So your DNA is normally a double strand like this, and so when you denature it in step one, it's going to basically separate into two individual templates. And then what we do is we develop some primers of a particular gene area that we want to make multiple copies of. And then we anneal them to these templates. Basically, we let them zipper up to these templates. That's called annealing. And so these primers, basically what they'll do is they'll kind of zipper up to their areas that are supposed to be complementary to the piece that we want to amplify. And these will give us our 3' OHs so that we can build off of them. And then the last piece is extension, where we add a polymerase that can basically fill in the rest. And so now we have 2 molecules instead of 1. And so if we keep looping that around in multiple cycles, then we can double with every generation. So for instance, we start off with 1 molecule. So basically here's our 2 strands that are separated. We prime them with our primer. That gives us our 3' OH, and then we extend them and fill in that gap. So now we have 2 molecules. And then if we send this back through another denaturation, then we'll have 4, and then we'll have 8, and so forth. And so eventually we'll just keep doubling, and we'll generate millions and millions of copies of DNA. PCR is a critical technique, especially before we're able to do any kind of analysis on DNA. So whenever we're doing DNA fingerprinting or DNA profiling, we have to amplify that DNA with PCR. And then the way we analyze the fragments of DNA that we is through a process called gel electrophoresis, which is kind of like a jello. It's like a little pad of jello that separates the DNA by size. And so the patterns that we see in the DNA, if they match, basically came from the same source. And so when you DNA fingerprint yourself, the important thing to remember is that your DNA fingerprints or your fragments come from only one of 2 people, your mom or your dad. And so as a result, because we all have different moms and dads and different combinations of moms and dads, then we have kind of a unique fragment pattern, just like we have unique traits even from our siblings, even though we have the same parents. And so DNA fingerprinting is used basically to identify whether or not we are related to somebody or whether we were somewhere, like if it's a forensic sort of a thing. So this is one way we can use DNA fingerprinting for test for viral infection. For a genetic disorder, we can pick up people who have genetic disorders or cancer perhaps, because there are very specific patterns that we look for in this particular process. So we can compare this to blood samples if we do blood typing as well. And we can identify the remains of bodies, for instance, by using this. So for instance, if we have a body that was like maybe burned beyond recognition, we can amplify some of that DNA and fingerprint it, and then we can determine whether or not that person is, who that person was. Okay, so for identification. So this is kind of what it looks like with crime scene. So here you can see you've got a marker, which is just our standard, just known sizes and fragments to give us a sense of reference point. We have a sample from the crime scene that we're able to generate 2 fragments from, and we have 2 suspects, suspect A and suspect B. So the fact that suspect B matches a crime scene, it suggests that suspect B did the crime. Or at least at the crime scene. Not necessarily did the crime, but was at least at the crime scene. So, and that's kind of how we use DNA analysis, especially in kind of legal parlance. Now, cloning is a little bit different. So basically, cloning is when we produce genetically identical copies of DNA, cells, or even organisms. So gene cloning is essentially when we're making many copies of a gene. So you could argue that polymerase chain reaction was a form of gene cloning. Recombinant DNA technology, however, is going to be a situation where you take DNA from one source and you put it into another. So for instance, if you create this rDNA, recombinant DNA, a vector, which is a structure that carries your gene, will introduce your gene of interest into a host, like for instance, a bacterium. And so basically what you're doing is you're putting in putting in a non-bacterial gene into a bacteria. It's kind of like transgenics. Okay. Now, the common vector we have is usually a plasmid. And a plasmid, remember, is that little ring of DNA that's part of the bacterial genome. And they're quite handy, actually, to be able to move back and forth. So this is kind of what it looks like. This is your basic recombinational history. Let's imagine we have a human gene. So we're able to cut out this human gene, and we have a vector, a plasmid vector. And then what we can do is we can basically add that insulin gene in this particular case to our plasmid vector. So now we have a recombinant plasmid that is carrying our gene of interest. And we can basically introduce that new vector, that new plasmid, into bacterium. And then from here on out, the bacteria will basically propagate and amplify our gene of interest. It will also make insulin. So we can either clone the gene by allowing the bacteria to propagate the gene, or we can have the gene make insulin. Both of those can be quite useful. So how do you go through gene cloning? Well, the first step is basically to use a restriction enzyme. And in this case, you're going to cleave both human and plasmid DNA. Now, the good thing is plasmids are natural things in bacteria, so they know what to do with that. And so during the cloning piece, it creates a gap in your plasmid DNA like we saw previously. And then we're going to pop in or insert our foreign DNA, in this case, our human DNA, into that particular area. Now, in order to do that, you have to use the same restriction enzyme because they create the same cutting pattern. So that allows you to basically insert the DNA into the plasmid. So this is kind of what it looks like. So you kind of cut it. Use a restriction enzyme. For instance, if you cut between AA and TT, two different ends, right? But in this case, you cut all the way through. So it's going to create two ends, right? Sticky ends. That unpaired ends, AATT, are unpaired ends. And so what's going to happen then is these sticky ends will be how you're going to attach your piece. Now, DNA ligase is going to be used to basically seal your insert into the plasmid. And so once your insert, which should have matching sticky ends to your plasmid, they basically lock into place, and then your ligase will seal them up. And then ultimately, you can introduce that to bacteria. Some of the bacterial cells will take up your plasmid. And then once the plasmid starts to replicate on its own, then your cloning has been successful. And so you can also use this to induce the bacteria to make the product that they could not make before. And so now they have kind of a new type of capacity. So what does it take for this gene now, this recombinant gene, to be expressed in bacteria? Well, first of all, it has to have the bacterial regulatory regions. So the bacteria has to recognize it in order to do that. So for instance, the gene that we put in there, it can't have introns because bacteria don't have any splicing. That's a eukaryotic thing. That's not a bacterial thing. So you can't put introns in there. It won't know what to do with it. It'll just make the introns and it'll just be nonsense. And so the other thing, in order to get rid of these, what we do is we kind of do what's called a cDNA production. So we oftentimes will use what's called reverse transcriptase to make one that lacks introns. So for instance, if we have an mRNA of our gene, right? Remember, our mRNA is going to have our 5' cap. It's going to have our open reading frame. That's a continuous reading frame with a start and a stop. And it's going to have our 3' tail. So what we do is we take this in a process called reverse transcription. And this reverse transcriptase is able to turn this into what's called a cDNA, a clonal DNA, which is a DNA version of that area. And so this is basically a DNA copy or a DNA version. So reverse transcriptase will do that. It'll make a DNA copy. of the RNA. And that'll give you a reading frame without enzyme. That you can put in. So basically, you'll make this clonal DNA or cDNA, a copy or complementary DNA. And so this will allow the gene, the bacteria, to recognize it and to know what to do with it. And so you can make a new product with this. In this case, it's insulin. It's new for the bacteria. It's not for us, right? Because we make insulin. Now, what about editing? Editing is another important one, right? This is basically where you target a specific sequence of DNA for either removal or replacement, right? So CRISPR is the big one of the day. So everybody's talking about CRISPR. This is basically clustered regularly interspaced short palindromic repeats. Say that one fast 5 times. And so this is a technology we've had now for a while. This isn't new. But it uses an enzyme set that comes from bacteria called Cas9. Cas9 has the ability to identify specific nucleotides to be cut. And what it uses is a guide RNA molecule to kind of help it identify a particular area. And so if we know a particular area, then what we can do is we can make a complementary RNA strand that can be used as a guide by Cas9. This will produce a break in the DNA, and it'll cause it to replace it. So it's kind of what Cas looks like. So here's basically our complementary target region, our guide RNA, so to speak, right? And this is going after our target gene. So what's going to happen is in Cas9, it's going to bring the guide RNA in. It's going to identify our gene of interest. And when it does that, it basically cuts out the gene and then replaces it with the new piece. So where you had a mutation in a genetic disorder before, then the new piece of DNA that it basically swaps in there, it cuts out the old one and puts in the new one, will have no mutation in it now. So the reason why we're excited about CRISPR is it has the capacity to actually be able to go in and fix genetic disorders, which is something we've never been able to do before. For instance, we can mute genetic disorders like sickle cell and cancer. We can get in there and fix these things. And so we can also allow us to be able to develop tests for things like COVID, right, which is SARS-CoV-2. So sickle cell, one of the things that can be done, obviously, is if you're fighting sickle cell, you're basically replacing the mutation with a new one. You could do that in the bone marrow. That'll give you good cells in the bone marrow to basically make new, fresh, healthy blood cells with. For cancer cells, essentially, you can make cells more efficient at fighting cancer. And you can reinsert those back into the bone marrow. Editing sperm. So you can remove a mutation from the sperm that would maybe cause cancer. And then ultimately, you would have IVF or in vitro fertilization therapy upon in vitro fertilization. So you can see how we can go in there, we can actually edit these different things. And now this is kind of where a lot of the pushback comes from, is because a lot of people see this as sort of designer kids, right? And a lot of people are worried that this will create a more high-tech version of eugenics, which is kind of what the Nazis were doing in terms of, you know, engineering people, basically. So some of the products we see are like, for instance, cells that can be genetically engineered. And so we just basically alter their DNA so they can basically produce a specific substance. So we looked at the bacteria was a good example of this one. This is kind of where our GMO situation comes from. So essentially, this is where the genome has been modified. And it's usually using the recombinant DNA strategy. And so that adds some sort of a new gene or new product that this thing did not have. Like, for instance, plants that can make a pesticide, for instance, as an example. Transgenic organisms, these are basically organisms that have a foreign gene inserted into their genome. And so there's also a lot of controversy about this as well, but we've been making transgenic organisms for a long time. I mean, as soon as we talk about adding a plasmid into a bacteria, that's a transgenic organism. So we've been using and making transgenic organisms for decades now. The only thing that's changed is nobody has ever proposed the idea of even modifying or doing anything like that to humans. So that's kind of where the controversy is coming in. So we use transgenic bacteria, and we have those. We have big vats of those. We call those bioreactors. Ultimately, these bacteria can be engineered to eat oil. And so that kind of helps clean up beaches after oil spills and things like that. We oftentimes will give these guys what's called suicide genes. So when they're done, they self-destruct, right? This basically controls them. And this is supposed to control them from going rogue. So we have frost minus or frost resistant bacteria that will help strawberries resist frost, right? Those are things like that. We have bacteria colonizing in the roots of corn, right? That's helping to produce insect toxins that will help corn resist insect damage. If you take a look at common produce, most of our corn, potato, soybean, and cotton plants have been engineered. They've either been engineered to be resistant to herbicides or insects or both. So engineering is a common thing. We've been doing it for a long time. It's not a new thing, right? So this is helping us to sort of decrease spoilage. For instance, the gene that causes browning in apples, if you knock that gene out, that increases shelf life. Now notice, you didn't do anything with this apple. So you didn't add anything in this apple. If you knock a gene out, basically this is just like a mutation. So it's just lost that ability to brown the apples. So you have to be careful about just, not every molecular advancement is an evil kind of a GMO sort of a thing. So you have to really look carefully at it, and you have to think thoughtfully about it, about what some of the downstream consequences of each of these things are. And for some of them, there's not that much. For instance, knocking out a gene in an apple is not that big of a deal. It's not going to hurt anybody. Maybe adding some crazy genes in there that can turn on herbicides and different types of toxins and things like that, maybe that one's a little more concerning. Maybe a bacteria that basically eats oil and has to have a suicide gene in it, maybe that one's a little concerning, because the question would be, what happens if the gene mutates that suicide gene, right? And all of a sudden the bacteria don't have the suicide gene. What happens if they live forever? We're going to have a world full of oil-eating bacteria. What does that look like? Right. So this is an example. So here's the apples with the mutation in it, and this is the normal apples. So notice this is not, I would not call this an engineering thing. Not engineering. This is just basic genetics. You could get a mutation of this apple by just waiting for it to happen. Right. So when you knock this out, it's basically just a genetic sort of a thing. It's not necessarily, you're not doing some crazy engineering thing. So some of the modified plants we have, so for instance, soybeans, have been engineered to produce fatty acids, which improves health. So it's a good thing, right? So leaves can be engineered to lose less water, which is helpful, less stressful in the plant, and also take in more carbon dioxide, which helps the environment. So that's a good thing, right? So it helps the crops to grow well in lots of different environments. Some plants can produce hormones, some can produce clotting factors, antibodies, vaccines. So this is basically using, I would argue that this is using technology to benefit our health. So not all GMO stuff is scary and evil. We actually have a lot of it out there that's giving us a lot of benefits. So of course, animals can also be modified to increase their food value. For instance, salmon that are transgenic, they can produce a growth hormone that causes them to grow faster, but they're engineered to be sterile, so they don't reproduce and take over. So if it causes faster growth, then this creates greater yield. And you can feed more people. So a transgenic mosquito, for instance, like that carries Zika virus, which is a problem, can produce a protein that essentially kills their offspring. So that's kind of like a viral transmission thing. So that's a good thing. So of course, here's your salmon and your mosquito. Now, gene farming, right? This is basically a situation where, like with the bacterial example, we use transgenic animals to produce pharmaceuticals in kind of the same way we did with the bacteria, right? So for instance, the genes that code for these proteins are basically incorporated in the DNA by recombinant DNA technology. And then they appear in the milk. Then you can use cloning to make many animals to produce the same product. And so you essentially get this mass production of this particular pharmaceutical that, you know, can have different things in it. So for instance, milk that would have antibiotics in it or vaccines in it, or it could have just a higher nutritional value, right? So this is one of those ideas of producing pharmaceuticals. So this is kind of what it looks like. So essentially, you would have to take a gene, like a growth hormone gene, and inject it into, like in this case, a goat, right? So this is like a donor egg. And then the transgenic goat essentially would make the human growth hormone, and that would basically have your milk with growth hormone in it. And then this, you can then develop the host goat to create multiple goats with this transgenic capacity. Now, I don't know how much this is used, but it is something that we can do. I think there would probably be a lot of pushback on that from a lot of people because there already is a lot of pushback on using hormones and things like that for milk production. But it is something that we can do. We can do it for a positive purpose. Mouse models are necessary for studying human diseases. If we didn't have mouse models, then we would not have the therapies that we have today. For instance, the cystic fibrosis allele, we have inserted that into mice. So basically, we've created cystic fibrosis mice. And so this allows us to be able to study these mice. They carry the same genes as the humans. And then what this basically allows us to do is understand basically how this is developing in mice. And the idea is if we can understand it in mice, we can then maybe provide something for humans. And so this also leads to the idea of xenotransplantation, which is transplantation of animal organs instead of human organs, like for instance, pig valves in heart transplants. So using an animal organ to basically replace one of our own. And so then we have the development of the Human Genome Project, which opens up the entire field of genomics, which is the study of the genome or genes. And the Human Genome Project in the early 2000s basically sort of revolutionized genetics as we understand it. For the first time, we have the entire human genome sequenced. And so we have, 3 billion bases. And so about 20,000-ish or so functional genes in humans that are actually on us all, at a given time. So the size of the genome is not proportional to the number of genes or the complexity of the organism. So for instance, less than 2% of the entire genome codes for functional proteins. 98% of it may be RNA molecules or something else. So in functional genomics, basically we look at how our genes function. And so what we look at is the roles of intergenic or non-coding DNA sequences in producing like things like small RNA and things like that. And so we look a lot at these sort of non-protein types of functions of DNA. That's all in there, right? Comparative genomics basically is looking at different things, like for instance, comparing the human genome to that of other animals. So for instance, a lot of times we'll use comparative genomics when we're looking at evolution. So for instance, how do two different organisms, genomes change the way they evolve? For instance, we're always talking about chimpanzees and humans, where we have our between 95 to 98% of our DNA sequences are alike. And so essentially the differences between the two is some, 2 to 5%. And so our genome to mice is about 85% similar, which is important because this allows us then to transfer knowledge used in mouse studies to understand humans better, because we're more alike than different. So the field of proteomics is essentially the study of the structure, function, and pretty much everything associated with proteins, right? And so we have a term for that. Essentially the lump sum of all the proteins in the cell. And so one gene, for instance, can code for several hundred different proteins. That's through alternative splicing. We talked about that earlier. And other mechanisms, but alternative splicing is a big one. And we're now just at a point where we can use 3D modeling, computer modeling, to understand protein structure much more. Now, bioinformatics is basically using a computer to study the genome, so and analyzing different types of patterns in genomic data and things of that nature. So we're still kind of picking through all that information that we just got about 20 years ago or so through the Human Genome Project. It's also led us to an idea of what's called gene therapy, which is kind of like CRISPR, is associated with the insertion of genetic material into cells for the treatment of a disorder. For instance, if you give the cell a healthy gene through gene therapy, then it can essentially use that healthy gene and fix the disorder. But it doesn't actually replace the bad gene. You're just adding a new one. So this can be successful in treating some things, for instance, inherited blindness, possibly. Things outside the body versus inside the body, ex vivo versus in vivo. Gene therapy methods are typically used. The ex vivo is a situation where we take stem cells that are removed. We talked a little bit about this a few chapters ago. So you remove some stem cells, a vector is added to those stem cells for the normal gene in question, and then you put the cells back into the patient. And so this has been successful for SCIDs, so severe combined immunodeficiency disease, hemophilia A, Alzheimer's, Parkinson's, Crohn's, and a couple of cancers this has been successful with. So in this particular case, like for instance, you will remove some bone marrow cells, then you'll incorporate, in this case, through a retroviral vector, a piece of normal gene into the cell itself, and then you'll put the newly rescued cell back into the patient, and hopefully this is the cell that will then take over its production. So in vivo, however, is basically where you inject DNA straight into the body. So you basically bundle it up in a virus, and it'll basically carry the gene itself. So if we've done this with patients with cystic fibrosis who lack a gene for the chloride transporter, and so we give it a functioning gene, either sprayed into the nose or injected into the respiratory tract, and they don't typically last very long because it's not actually part of the cell itself, but it's not very invasive. It's an easy one to do, can give you quick results, and it could be used for small, like temporary sorts of things, cardiovascular disease, endocrine disorders, Huntington's disease, just something to give you a little bit of short-term coverage. But we continually are working on all these different types of therapeutics. So that is the end of chapter 22.